Hello, everybody. This is another episode of the Wizards of Ox Live, and welcome. And as every Monday there's four of us, four wizards from different parts of the world, helping you with your low oxalate journey. And so let me just introduce you. We have Monique Attinger at my left, I guess. I don't think everybody sees us the same way.
It might be according to my recording, which who knows? Anyway.
Okay, but anyway, Monique Attinger from Canada. We have Susan Owens from Houston this time, from Houston in Texas. And we have Carla Wiersma still talking to us from beautiful, cold Colorado. I'm Patricia de la Garza, and I'm talking to you from Brussels in Belgium. It's already night here, and we are going to get started. Today, we're going to be talking about some of those questions you have been asking, and you've been wondering why they're not already in the page. And basically we have a log, and we have like a previous meeting with also the moderators, and and everybody takes a question that they feel comfortable answering, because since everybody's coming from a different oxalate level, a one-size answer is a very risky thing. We could be telling you something that would be perfectly fine if you were already on a low oxalate lifestyle since years, but might not be the right thing if you're just starting and you're coming from 3,000 milligrams a day or more, or if you have endogenous oxalate production. So we try to really be very conscientious and look at the science, document everything, and that takes time. So today, what we're going to do is that we're going to take some of those questions and we're going to be answering it for you, okay? So if you want to get us started, I guess, Monique? Oh, well,
apparently I was muted. Sorry about that. Susan has done some work in putting together some information, both on our question, which has to do with UTI, and we also have a question which is dealing with ferritin. So let me just read our UTI question, and this will then go up on our Trying Low Oxalates page, and we will have our video attached to it. So everyone will know that it's been answered and we've got some information there for it. The question is, this is my second time in the past 3 months to have a major UTI and major UTI symptoms. Can oxalate cause infection or just urinary tract discomfort? I hesitate to take antibiotics due to gut health, but I'm afraid not to as well. Is D-mannose a good option, or is that more of a preventative? And maybe we can tackle that for this particular member.
Yes, and before I forget, please remember that whatever we say, it should not be taken as medical advice. We're sharing the science and we're sharing our own experiences. So we are assuming that you are under care of a caring doctor.
Thank you for that. Thank you for that reminder, because, what's different about a physician is supposedly, we hope that he is keeping track of you and knows what all the different issues are that you're working with all at the same time. And so there's no way that we can know that, because we're going to be specifically talking about one little area, but how it fits in the big picture is really important. But also, we may be talking about science that your doctor hasn't had the time to read yet. And so we encourage you to have very good discussions with your doctor. And if you learn something here, get the documentation, get the studies. Don't say that these, I heard it on the internet, because that's not going to go over. What may give you a little bit more clout is if you can bring in some scientific studies that say such and such and that sort of thing. So just warn that, and then you have to know your own physician, because some physicians are more open to, outside information than others are. And if your doctor doesn't match you in that way, then see if you can find somebody in your community who's a closer match who will talk to you about these kinds of things. So anyway, I just wanted to put that warning out because, not everybody is a good match with everybody else, even on a professional level. So we just want to make sure that you're getting a place where you are comfortable dialoguing with your physician. Does that make sense? Really important. You turned your thing on
really,
again?
I was muted again. Really good points, Susan, that people need to have a physician that they can really work with. And everyone will have their own preferences on how that relationship should proceed. But I really like somebody who works with me as a partner and where I can take that kind of information in. So it's a great reminder of, finding the right person for you, and then, really making sure that you have somebody who understands what your health status is and can help you with that. Yeah.
And also that doesn't make you feel like you're like 2 inches tall, and they're a giant. You don't want to be in a situation. Nobody wants to be in that kind of situation.
Absolutely. But the UTI, yeah.
Huh?
No, but the UTI question is one that comes back again and again and again.
Oh gosh, we have heard that so many times, but I don't want you to think that we were the first people to hear this story, because our project, and I started the project many years ago with some help, but it started with a lady talking to somebody at the Autism Research Institute who was wondering if some of the pain issues in autism could be caused by oxalate. And that was something that had been explored by the Vulvar Pain Foundation. And this particular lady had vulvar pain and had utilized the information from the Vulvar Pain Foundation, and let's see.
Found it useful. Yeah.
And found it useful. And so I'm going to do a screen share here so you can see this. Oh, host disabled participant screen sharing.
You're able to share now, Susan. I've got it set up.
Okay, thank you. All right, share. So this is their logo. This is the VP Foundation. And let me do just a slideshow thing here. One slide. There we go. Anyway, so I want to tell you about what happened with the Vulvar Pain Foundation. This project, their foundation is probably 40 years old. So they've been doing this for a long, long time. And they started with the work of a researcher named Dr. Clive Solomons. And what Dr. Solomons did was started what he called the Pain Project. And he was basically somebody who was investigating changes in connective tissue in women who had vulvar pain. And so, and that's your lady area, basically, if you're not familiar with that term. And anyway, so one of the things that he did was he got to be very proficient in measuring oxalate. And so he had the very unusual idea of, instead of doing a 24-hour collection, which is really kind of the standard protocol that has been used for kidney stone disease. And what he did was he would have people keep a diary on the day that they collected urine, and they would collect a tube of urine every time they went to the bathroom. And so on that day, they would write down, this hurts, that hurts, I have a migraine headache, just whatever was going on that day. And so this, of course, would be blinded to Dr. Solomons. He would know about this when he was doing the measurement of how much oxalate level was there. And so they had, decades worth of this sort of data on these women with autism, I mean, with vulvar pain. And so one of the things that they did is then after they got the results back of when the oxalate was elevated, then they would do a comparison of what the women said was going on that day and how much oxalate was there, and look at the rhythms that were there over the day. And even in some women, you know, they went day to day to see whether they had the same pattern from day to day. And so he did really a lot of excellent work. Now, at the beginning of our project, we utilized that lab to measure children with autism. And so I ended up talking to not Dr. Solomons, but his wife, and learned that early in his research, and I don't even think it was when he was looking at oxalate, he got kind of burned in the submitting your papers for approval and that sort of thing. And he just decided that it wasn't worth the pain of going through that kind of process. And so he was really happy with just working within the domains of the Vulvar Pain Foundation. Now, there are plenty of doctors that treat vulvar pain that have a lot of respect for him. But if you just go by what he published in the literature, there's only one study out there. So I wanted to warn you that if you go looking for this, you won't find a whole lot of tracks. But there's a reason for the lack of tracks, is that Dr. Solomons invested himself in the Vulvar Pain Foundation. You can find the information there. And if you do have vulvar pain, we very much encourage you to go there, because they have a lot of experience with it. And they do a lot of coaching of, you know, one woman to another woman kind of thing. And that's very valuable. So what they found out, though, was that a lot of their women would get what they thought was UTIs. And sometimes they would get a UTI and it would be culture negative. And another time she would get a UTI and it would be culture positive. And so, you know, the general way it works is you go to the doctor and you think you have a UTI, and so he takes a sample and he sends it off to the lab, and maybe 2 or 3 days later, you'll get results, and they'll either say you did have an infection or you didn't have an infection, but they never measure oxalate. Well, that wasn't the case with Dr. Solomons, because they would take split samples, and, you know, one sample would go to the laboratory to get measured for a urinary tract infection, and the other one would go to Dr. Solomons, who would measure the oxalate. And so what they found out over decades of work is that the women could not tell the difference symptomatically from a UTI that had bacteria in the urine and one that didn't. Is that shocking? What do you think, ladies?
I'm not shocked, because I'm very familiar with that study, and I thought it was amazing and so smart to measure every time you went to the bathroom, because then you have a correlation with that particular thing. So the dumping cycles or the dumping moments, you could actually map out, you know, with whatever was happening. But many times I felt like a UTI, and it hasn't been the case. So,
well, because you got the results back from the lab and it was negative.
Exactly.
Now there are some doctors out there, and I've heard this from many people, that the doctor said, oh, well, that doesn't make any difference because they're not measuring it in the tissue and blah, 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 blah. And then they'll tell you, oh, in order to get rid of your infection, we're going to need to treat you with, you know, some elaborate protocol that costs $1 jillion. And so, but they're not looking at the oxalate angle. And so, you know, it's going to take time to get these different points of view kind of sorted out. And so I'm not pretending that they are sorted out yet. But what I am saying is that definitely the women that sometimes had positive UTIs and sometimes had negative UTIs, couldn't tell the difference. In other words, on the front end, you can't tell the difference. So it feels exactly the same. Now, what happened later, and this is after even Dr. Solomon, well, let me show Dr. Solomon's. I have his picture up here, page down. Yeah, there he is. And this is the pain project that he had. He was the director of that, and he died, I don't know, maybe about 5 years ago, I would guess. But anyway, so it was later that it was discovered that the bacteria that is like 85 or 90% of the infections that they find in UTIs is E. coli. And some of you may hear that, you know, and that's maybe the only bacterium, you know, whose name you've ever recognized. But anyway, what they found is that E. coli, there was a study in China, and they were looking at the genes that degrade oxalate and which bacteria degrade oxalate. And guess what? It is E. coli that is the primary, like, bacteria that might be there in the urine to degrade oxalate. Now, see, this is a very different way of looking at the world, because if you think, oh, it's an invader, it's a bad guy. And I have this little story that I tell, and I think it's useful to some people to be thinking about this. But let's just pretend that I am a Martian and I am driving over a convenience store, and I look down and I see a fire truck and an ambulance, and I see 5 police cars, and they all have their sirens going and their lights flashing, and you're looking at it and going, I don't know anything about American society, but I think that's bad. And so from my spaceship, I'm gonna zap and blow up the fire truck, blow up the ambulance, and blow up the 5 police cars. Why? Because I don't understand what's going on. Now, if you, on the other hand, were somebody that was tootling around the street thinking, oh, I want to go in and get a bag of chips, you're going to drive by there, and when you see all those emergency vehicles outside, you're going to say, not going there. I don't want to get shot. I think I'm going to go somewhere else and get my chips, right? So this is what I'm saying. We in science right now, we spent so many years with this way of looking at the universe that is, you know, the idea that the way you get to the top is by killing everything underneath you, everything that's lesser than you are. And this is the Darwinian thinking, survival of the fittest. And so we drag that whole concept into medicine and in the way that we started looking at bacteria. And so what happened is there was a major change in the research that occurred about 2002 and 2003 when the Human Genome Projects, you know, began, and they began analyzing. They had the technique called PCR to look at the bacteria and say, oh, okay, well, we didn't know how to figure it out by looking at it in a microscope, but we can look at the DNA, and then we can also look at the RNA. Now, what is the difference between those two things? RNA is the response to the environment. It's telling you that your certain genes are being utilized and they're making the proteins that come from the DNA. And so suddenly the microbiologists had the ability to look at what they hadn't been able to see. And so about 20 years ago, I was invited to hear a man who was introduced as one of the fathers of modern microbiology. And during our off time while I was there, he said, Susan, I want you to know something. We only are basing what we do on knowledge of 15% of the problem, because know how to culture certain things, but we don't know how to culture other things. So all of the techniques that our doctors were trained in were based on the science 20 years ago and earlier that was based on what we knew how to culture and what didn't mind being exposed to air. But now the scientists have techniques to study the ones that are not happy exposed to air. And so that's how we're getting the understanding now from all of this huge bit of work that it's not survival of the fittest in the gut. That's not how they operate. They operate more the I'll scratch you back, your back, you scratch mine. They're taking care of each other. They're taking care of environmental issues that show up. So, you know, as soon as something gets blown up, then they try to fix it. And they scramble and they increase their levels because they're the things that know how to do that work. And so this is a very different way of looking at tests like these culture tests or tests like GI map or something like that, because they're going to say, they're going to just, in your head, you're going to see the red letters that say high or low, and you're going to think, oh no, they're bad guys and we need to kill them. And that is working with science that's about 30 or 40 years old. And we need to grow up, you know, and start looking at the new work that the scientists are doing. So it might really be that E. coli is a bystander, and it's a bystander that actually is doing a job, like it's getting rid of the bad stuff. Because the bad stuff is really the oxalate that's accumulating in your urine that's going to cause kidney stones. So we might have the system inside of our bodies to get rid of the oxalate. And when it's high, then the E. coli level is going to go up and it's going to expand enough where somebody can measure it. See, that gives you a whole different way of looking at this whole deal, because your doctor is going to look at a positive culture. And what is he going to do, Monique?
Oh, for sure, you're going to end up on antibiotics. And really what you're saying here is that it's correlation, not causation. Oxalate's gone up in the urine, so E. coli's gone up in the urine. It's not that E. coli is doing the bad thing. And in fact, maybe even oxalate. Oxalate is the one that's driving that inflammation and the pain and symptoms that somebody's having when they think they have a UTI, or at least that's a possibility, right?
Well, right. And again, this needs to be clarified. And we are not the bench scientists that are going to do this clarification. This is other people that will do this work. I want to make that totally clear. But what I'm saying is that the Vulvar Pain Foundation found out women cannot tell the difference. So the pain may have been what Dr. Solomons was studying, that was also messing up the vulvar pain tissue, not just the urinary tract. You get me?
Well, for sure. I noticed there came a point when I was getting what felt like a UTI happening every month before my cycle. And so if I'd have, you know, I was willing to try other things to see if I could handle the inflammation rather than go and get another dose of antibiotics. And that was a very good decision, because it meant that I discovered that anti-inflammatories like anti-inflammatory supplements like MSM just made all the difference for me in terms of, you know, whether or not I felt like I had a UTI coming on or not.
What I want to say is what we didn't know back then also was that we are completely dependent on our microbes to get rid of oxalate. So what happens when you treat a urinary tract infection with an antibiotic? Any guess?
Takes out the good and the bad.
Yeah, it takes out everybody. Like it's
Just
not,
killing your friends. Yeah.
yeah, it's not
It's not
specific.
discriminate.
It is. Well, I mean, they are a bit like, you know, certain ones do certain, you know, kill a certain set and that kind of thing. But we now know through some work done in China that every different kind of bacteria, there's some of them that degrade oxalate. So it's a very broad thing that is, you know, over a large diversity of microbes. So, you know, what do doctors do? I mean, I've seen this happen, that a doctor will give an antibiotic and it won't work. And then they'll give another antibiotic and it won't work. And then they give another antibiotic. And by the end of it, they've given 6 or 7 different antibiotics because they're trying to target different microbes and they don't really know what's causing, you know, the difficulty. And that's why they keep switching it. And so if this is in your history, if you have a history of people, you know, trying to chase infections, you know, the wise thing is to take a broader look and see if maybe there's some other element like oxalate. I'm not saying it's always, you know, what's in there, but if it isn't working, then you know, it is appropriate to ask the question, why isn't it working? And, you know, what else could be going on? But this is how people get trapped, because I was very shocked when I went to a well woman visit, and they always take, you know, a little bit of pee and they test it for bacterial infection, and they found something and they sent it off to the lab and it came back positive for E. coli. So my doctor sent in a antibiotic prescription. This was a number of years ago. And so I said, okay, I guess I'll take it. And I took it. Now, I had no symptoms. You understand? This was a well woman visit. This was not a, I have a UTI visit. Okay. And so I took the antibiotic and it messed up my knees so bad that I could not go. I could not walk for about 6 weeks. That one pill. Okay. And so that motivated me to go look in the literature and see what happened, what just happened to me. And I found there was a huge literature that was talking about that a lot of scientists had figured out that if you have non-symptomatic UTIs, which is what are they going to do when you go to a well woman's visit? You have no symptoms, right? So anything they're going to find that way is basically going to be a non-symptomatic UTI. So they shouldn't be trying to treat it according to these scientists. The scientists found that if you treated the non-symptomatic UTI, what it would do is it would make it worse for you. So the time when you do have a quote, real UTI, then the antibiotic won't work because it's developed bacterial resistance. So there's a ton of literature on this issue. And I do think that the information has gotten to the doctors, because I've noticed that a lot of doctors are a little bit more reluctant to, you know, go from the well woman visit in the urine and getting a urinary tract infection and going on that way. So this is all, you know, I want to say you and I cannot be responsible for wherever the information exchange is going on between physicians and the scientists, but we can, thankfully, we have these great big databases where anybody can go and look up the study and you can see what the scientists have found. So this is just an area where maybe some caution is, especially if you know that you have had an oxalate problem, that you've been, you know, eating spinach smoothies and, you know, doing a lot of things that would raise your oxalate over a long period of time, and that you might be doing this thing where the oxalate is kind of leaving periodically and cyclically, and it might be that is what's triggering the UTI. And as you said, Monique, we've had so many women in our project tell us that their dumping symptoms are so much worse related to their monthly periods. Is that true of all of you ladies? No?
It was true for me eventually, but initially I didn't see a pattern like that, but over time it became obvious.
But have you heard from other women about that?
I do think I've heard from others who have mentioned that there does seem to be a pattern to where they dump that appears to be related to their cycle.
Right.
But with that said, some of them have been curious about what they can do to help there.
And this is what I want to talk about next, because there are some things that are in the medical literature that tell you what works, and that is not geared towards killing killing microbes that you might need to degrade the oxalate. So do you want to carry, who wants to carry that torch?
Well, we can start out by saying, because the question was asking about D-mannose. I think we have people who have used D-mannose here. Have you, Patricia?
Yes, I have. Actually, I just wanted to say, to think a little bit like that. Those of you that know me know that I'm a gardener, and we always talk about plants and weeds, even though they're all plants. But so who decides who's a plant and who's a weed? And why is each plant where it is, especially in a garden that hasn't been treated, you haven't planted it specifically. And when you are working into like understanding the ecosystems in a garden, when you start looking at specific weeds, those weeds are there for a reason. And actually they serve as diagnostic tools of what's happening in the soil and what the conditions of the soil are. So, if you have a very poor soil, you're going to have weeds that have deep tap roots, look like carrots that are meters long, that are going to go and get those nutrients way back when. And then you have some others that have roots that are like spider webs that are just trying to hold the ground because there's a risk for erosion. So my point here is just very quickly is that everything is there for a reason. So just like Susan says, you have an elevation of E. coli. Why do you have an elevation of E. coli? There's something in the substrate. There's some reason for it, that it has the right conditions to germinate, so to speak. And the more that you have of those nutrients, if you want to think of it that way, and in this case, if E. coli eats oxalate, then if you've seen a lot of E. coli, it's probably because there's a lot of food to eat and there's a lot of oxalate floating around. So you have to start looking at starting to change the substrate instead of going for the killing strategies. So in my gardening example, if you feed a soil that was poor, that has these really deep tap roots, and you put, some fertilizer and you put some compost on top and you compact the thing and you do all these things, you're going to see that those weeds disappear on their own. And it's not because you killed them or you pulled them or you did anything about it, is that the germination conditions changed. So they don't have a reason to be there anymore. So if you start thinking of it like that, you start thinking of your gut and your private parts and your nose and your sinuses and everything as a garden, and then you start understanding all these relationships better. So what happens? So, some of the things that you can do is, for example, change the pH, because many times bacteria, their germination conditions are very restricted to certain pH. So, that's why some people, some old wives' tales talk about, having some diluted vinegar and wash it around there, doing all these different things that just change those conditions. D-mannose helps. Cranberry juice helps because it changes all these conditions. So,
But I'd also like to say, as somebody who was having like every month the symptoms of a UTI, I've found that MSM and methionine both helped. And the research actually points to methionine as a support where you can take that in situations of UTI symptoms. And they actually tested it with pregnant women who they didn't want to necessarily be taking antibiotics. And they would give them the methionine with cranberry juice and D-mannose first as one group. And then there was another group that would get antibiotics. And then there was a third group that got antibiotics plus methionine. And they found no downside to taking methionine, no matter what group you're in, which is not a surprise to me, because if it's taking down the inflammation, that's what's really dealing with the symptoms. And so my own experience with adding, supplements like methionine or MSM is that I could get out in front of this kind of symptom where, my system was irritated, I'm having pain, I'm having sense of urgency, I'm having all these things, and have it be completely resolved in, a day or two with an anti-inflammatory methyl donor like MSM or like methionine. And MSM worked for me just as well as methionine did. So I've used both.
Well, maybe it's just never been studied like that.
Yeah, it's possible,
and they need
isn't
to
it?
do
Yeah.
that,
It's possible. Yeah. So maybe I was just an N=1 experiment there. So.
Yeah. But I think what's important here is to realize that, we know what we know today. And if you go look something up in PubMed, you may find nothing in some area that you're looking up. And that doesn't mean it's a bad idea. It just means nobody studied it yet. And so maybe if you look 5 years from now, you will find a study that's looking at exactly what you thought anyway. And so we're stuck to where we are. The scientists do their job, and that's a wonderful job. And we need to know what the differences are between this profession and that profession and what you go for each thing. But we do know that the scientists are really on top of this. As far as if you talk to microbiologists now, they will tell you that taking antibiotics is really, really dangerous in the sense that you don't know how it's going to modify all of the microbes, all of your body, not just in your gastrointestinal tract, but on your skin and just everywhere else. And so I don't know if any of you have anything else to say about that, but it's just a time of rapid changes about these ideas. And we are not responsible for these rapid changes. We're just watching them and trying to be instructed by them, and not just trying to get all of our information from, just one place. We want to get it from the diversity of the professional people that have looked at this area.
Well, maybe
I hope that makes sense.
That makes perfect sense. And so I'm thinking maybe with that, we can spend a bit of time, because we've actually spent a fair amount of time on this question, but maybe we can spend a bit of time on our iron question. And that question is, I have low iron, low saturation, and low ferritin, despite supplementing with both iron and eating meat. Folate, folate, vitamin D, and B12 are all normal, and this person has not been diagnosed as a celiac. There is some indication that vitamin C with meat can increase absorption of iron, and this person's wondering why this is, and what she can do to really handle this issue with low iron and low ferritin, despite all the efforts that she's making. And I know that we've got some information on this too, so I'd love to get this out, because I think a lot of us have had issues with low iron. I've actually had to have iron infusions in order to deal with really low ferritin. So I think it's a great topic for us to talk about.
Okay, can you still see my screen?
Yes.
Great. Okay. What again, I want to emphasize that we are stuck at wherever the scientists are in figuring this all out. And so they're not through. They're in the process of looking at a lot of these things with new eyes. And so, first of all, I'm going to acquaint you with iron oxalate. Did you know that it looks like that? And you can think about, when you think about rust, what color is it?
Orange. Yeah.
Yeah.
Or red. Yeah.
So that shouldn't surprise you what that looks like. Now, some instructors figured, some chemistry instructors figured out what the structure was for this iron oxalate. You can see the iron there in the center, and you can see this is oxalate. It has 2 carbons that are connected to each other, surrounded by 4 oxygens. And you can see how the oxygens that don't have the double bond have the negative charge that is attracted to the iron. And so you can see how this coordinates. So there are 3, they're blue, red, and green that make up the oxalate. So we don't know very much about how often this is formed in the body when we're high oxalate. So it may be that a lot of that iron is trapped, but they really haven't looked at it that way. And why would that be? That the reason for that is that suspicions about a role for oxalate has not been considered for most biochemistry, because oxalate was only thought to be an issue in the kidney, where high levels of calcium, a different mineral, and high levels of oxalate can lead to kidney stones. And so it's dramatic. Look at this. This is a ruler, and this is showing you some calcium oxalate stones. And this is also a calcium oxalate stone. And you can see how big that thing is.
That's huge.
Isn't that amazing?
That is
Painful.
huge.
So obviously this is like, the elephant in the living room. This is something that you really notice. So the people that ended up running the oxalate field were kidney doctors. And when I went to my first oxalate conference, I was the only person there that was not a kidney doctor or involved with the kidney somehow. And so things began to change when scientists discovered that sulfate and oxalate anions, that's negatively charged ions, trade places with each other. They do this to balance the concentrations of themselves in a short list of anions, negatively charged ions, that move to opposite sides of a cell membrane using these transporters. So this is just showing you that this work was done, I don't know, maybe about 10 years ago. No, more than that, maybe 15 years ago. But this is why this is a new area, and why people who went to medical school 20 years ago wouldn't have heard about this, because the work hadn't been done at that point. So therefore, what does this have to do with transferrin? Well, transferrin is a transporter for iron. And this is a picture of what it looks like. But what they discovered in about the last 10 years or so was that iron and transferrin is coordinated or joined to the negatively charged ion or anion carbonate or the anion oxalate. So the bond to oxalate is so firm that it won't let go. So the job of this molecule is to grab up iron and then release it into the inside of your cell. But if oxalate gets there first, it can't do that, and it won't do that. And so so now there's some laboratories trying to develop techniques to discover like who has this problem and who doesn't. And it's not yet like mainstream. It's more academia that is looking at this. But, I hope that it will eventually transfer into ordinary practice. But what does this mean? Well, this is from a paper that was looking at how this works. And so you can see transferrin, when it doesn't have iron attached to it, it's called apotransferrin. But once you add the iron to it, it becomes holotransferrin. And then it goes into the cell, it dumps off its iron, and then your cell can use the iron. And then the empty one goes back to the cell surface and loses that iron. Okay, so some of the iron is used inside the cell, and some of it looks like some right here, they're showing some iron leaving outside the cell too. This is a quote from this study, cellular iron uptake, trafficking, and metabolism. The iron is then transported across the endosomal membrane by a transporter called DMT1, where it becomes part of the poorly characterized, that means they don't know very much about it, labile iron pool. So that's what the LIP, that's not for lipid. Iron in this iron pool acts as an intermediate and can be utilized for storage in the iron storage protein ferritin. Okay, or it can be used for synthesis of heme. You've heard of hemoglobin, it has heme inside of it, and iron sulfur clusters that are huge, very important for mitochondrial function, or it can even go into the cytosol. So iron can also be exported from the cell by a molecule called ferroportin. So it's really complicated, and there are lots of different places for it to go. This is for another one from that study, and they're showing this frataxin. You may have heard of a condition called Friedreich's ataxia, but it is a problem with this molecule right here. And so it interferes then with the trafficking of this iron inside your cell. And this shows that you end up with a buildup of iron inside your cell instead of where it needs to be. So just to clarify that, I have, there we go. This describes Friedreich's ataxia, and I encourage you to look at that if that has anything to do with your family or anything like that. But another thing is that transferrin also is very changed in how it functions by how that protein is glycosylated, which means whether it has these big carbohydrate chains on it. And so this is a picture of that. And then down here we have a study that is looking at the mass of it, depending on how much that glycosylation has occurred. So I'm just saying, telling you all this, so you can know that it's kind of complicated about all the different places that iron may just be disrupted. And actually, when one of those slides, I also showed that ascorbic acid can be really important. So you may hear some people say, oh, take ascorbic acid and that will help your iron chemistry. And it might. But it gets really complicated because also iron is something that can encourage the growth of bacteria. And so sometimes our bodies have this process going on where they are trying to kind of hide the iron, and they kind of stash it away, and they call this the iron deficiency anemia of chronic disease or chronic illness. And I don't know if anybody's ever dealt with that, but I did with my mother. And what happened is they kept giving her iron, trying to correct her anemia, and it just didn't correct. And I don't know how many people have had that happen to you. Has that happened to anybody else's family?
Not as bad as some, but I certainly, like, I had been doing all kinds of things before I actually took iron infusions, which included, trying to carefully construct meals. So I had a high vitamin C source like red peppers, and then I would have, a steak or something. And I would try to kind of amp up the amount that I was getting from diet at the same time as I would take a supplement. And yet I stayed very flat with iron. And so, I'd love to hear Carla's experience because she's had some of this too.
We had that with Ian. He had, when he was in the hospital with the strep infection, he had iron deficiency anemia. And at one point they had him on, I think it was 45 milligrams of iron to try to get those iron levels up, and didn't budge. We threw in lactoferrin,
Right.
and that actually helped a little bit, but it raised it a little bit and then nothing else. I went to a magnesium group and was asking, the question, how do you get the iron levels up when the supplements, just aren't doing it? And someone said, take a look at copper. Look at his copper levels. I bet you're going to find that his copper levels are right about the same as his iron levels. And he had actually given me a couple of studies. Took it to our pediatrician. Pediatrician went, hadn't seen this before. Let's give this a try. Stop supplementing with the iron. We'll give him some copper. We're going to, give him a little bit of zinc to balance that out, because we don't want to go overboard with the copper. We want to make sure things are balanced. A week later, his copper levels and his iron levels had raised considerably, not to the normal level, but enough to go, ooh, we're on the right track here. So we continued that for another week, and his copper levels and his iron levels normalized. They were at the low end of the normal range, but they were normal. And he'd been taking iron supplements for 45 days with nothing happening, and then just not doing the iron and doing the copper, seemed to get him where he needed to be. And, we kept that up for a little bit longer. And then we were able to slowly, get him off, to a smaller dose of copper that, he would have in like a multivitamin. And he's been fine ever since. So, is it one size fits all? No. There are some people who are high copper, so that may not be the answer. But if you're finding that all the iron supplementation isn't getting you anywhere, then copper might be something that you want to look at as a potential, help. It was for us.
Well, I want to point out that when I learned the most about this was when my mother was in a rehab center, and we realized that she had been on iron for over a year, and it had not corrected her anemia at all. Okay. And so finally I had a discussion with her doctor, and I said, if it hasn't worked in a year, maybe this isn't the right approach. And so we quit the iron, and then, we got busy with a lot of other things that we were working with and kind of forgot about it. And then the next time that I had her blood checked, it was normal. Like, it corrected, and we didn't do anything except take, wait a long time. I'm sure it was months between the times we measured it, but the anemia was gone. And so I think some people think that the only way to correct anemia is to give you a whole bunch of iron, and there are probably more alternatives out there, like as you were saying.
Well, and this is really interesting to me in hindsight, because yeah, I'd been on iron for ages after my daughter was born, and my ferritin was in the tank. It was just, I think I was at 15 or something, and yet my heme iron was okay, and it didn't seem to matter how much iron I took, I could not budge that ferritin number. But when they give you IV iron infusions, you're getting 300 milligrams of iron all at once. Like after the first one, I remember feeling really good, but then they want to give you 3 of them in a row to make sure that you're not going to drop down again. Nobody ever checked my copper or anything like that. And after the second one, I felt really bad. And I went, I'm not taking a third one, thanks. I think I'm good. So there's this funny sort of, if some is good, more is better kind of thing out there too. I mean, it's not just with, with antibiotics, it's like, if some is good, more is better, but it's also if too many bacteria are proliferating, it must automatically be bad, right? Like we seem to have this kind of thought process, which is I'm going to say kind of biased to overreacting to things, as opposed to just coming in at an appropriate level somehow. So certainly my experience with iron was that these iron infusions, one was okay. I felt good after the first one, but after that, boy, it was bad. So if somebody had checked my copper, maybe that would have been an easier fix right from the beginning.
Well, I mean, what we don't know is, tell me if this is not the truth. I think I know your story well enough to know that you felt like once you figured out that your kids had an oxalate problem, that you probably had a really big problem, and you started reducing oxalate yourself. And so it may have been, but back when you were dealing with the anemia, you also could have been high oxalate, although you probably didn't know about it back then.
Well, with the anemia, it was actually after we'd started on a low oxalate diet, but I'd also had 2 full-term pregnancies. I'd nursed 2 babies. I'd had 3 miscarriages in there. So my body had a fair amount that it was dealing with in about 8 years or so. And then we started doing low oxalate. Of course, all the oxalate's leaving your body, and you've got whatever oxalate's having fun doing as it's on its way out. So yeah, I think there were probably multiple factors there as to why I was so low in ferritin.
You don't know your oxalate status long ago. And
no, gosh, no. I tried to become a vegetarian more than once, so I can only guess what I was doing to myself.
Well, the thing is, it's going to be time before we have scientists who will investigate what happens, more generally to your iron chemistry when you're high oxalate. We know about this transferrin because that's been studied. Yeah. They already have scientists working on that end. But I think it would be foolish for us to say, oh, well, that's the only thing that happens, because there may be other processes going on, and we don't really know much about that, and there hasn't been much suspicion of that. So we'll wait and see. And once they find out something really good, then we'll certainly tell all our group members about it. But we can't invent work that's not there yet. We don't know what's really going on with some of this, but we can say that we've had a lot of experience with people correcting issues with anemia in different ways. And how many stories have we had about people getting over anemia after reducing oxalate? Do we even have a number for that, or a feeling?
I don't know that we've got a number, but I know that over the years we've certainly suggested various approaches to people. And Patricia, the lactoferrin, isn't that you that I learned that from?
Mm-hmm.
And basically it functions the same as the lactoferrin. It's just getting in a different way.
Well, and so, we do know that there are multiple ways that people can support their systems. And I think I recently saw that higher doses of iron, but more intermittently might work better for those who are trying to deal with an iron problem as well. So, oh, and we've got a comment from one of our folks who's online with us. That's what happened to me just recently within the last few months. I didn't supplement iron. All I did was lower oxalates, and then my anemia went away. There you go. There's another story
Yay!
we can add to our list of stories.
I don't think I had an update from her anyway, and I saw her name, and so I'm thrilled that that may have happened.
Yeah, this is great. It's great to hear stories where, there's so many of us actually who have good stories to tell about issues that we had that went away. But, this business of iron, gosh, my mother's got friends in their 80s who still take iron every day. And, I'm not privy to what their diet looks like, and I'm not privy to whether or not they've had any of their other nutrients checked. So, it's wonderful that we can at least help those who are on our group.
Well, I do think that there is also a copper deficiency that tends to happen when you get older. And, I don't know if doctors for very old people really look at this very much, but if you have a loved one and they're, in their 80s or 90s and they're having problems with anemia, definitely look at this copper thing that Carla mentioned, and get your doctor to check that out, because it's not only going to affect their iron level, but it may affect other things in their chemistry too, because copper is pretty important.
And I think the most important of it all is balance. You need to have everything in balance. So not one thing is bad, not one thing is good. You have to have a little bit of everything in the right proportions, and that's where health is found.
Well, as opposed to more is better.
Exactly. But one of the things that I want to point out is, when you get older, things just don't work as well. Just like if you've had an old car, that it's not like doing like it used to do, and you have to take it into the shop more often. Well, I'm getting old enough that I'm running into that myself. But what I'm trying to say is we have differences in how older people absorb and handle nutrients. And there are some doctors that specialize in this area, and I still have not actually gotten to know one who, whose practice is like this. But I mean, there's definitely ways that you can overcome issues that become nutritional just because you got older. Does that make sense?
Well, there's research which shows that we're not digesting the same way that we did when we were young. And one of the things I've noticed actually with my mom, who's in her 80s, is that she started to avoid protein. I don't think it's a good thing, but I think it's partially because she's not digesting as well. So, I will be making sure that she gets some digestive enzymes in her routine so that she can, like, if anything, as you get older, you need really good quality protein, and you probably need more of it than somebody who's young, because you're not absorbing it quite the same, may not be using it quite the same. So, yeah.
I remember with my mother-in-law that she had a kind of blood leukemia that's not supposed to kill you. It's chronic lymphocytic leukemia.
Oh, that sounds like what my father-in-law has, actually.
Oh, really?
Yeah.
Well, what was interesting is that she'd always been, kind of like, as one of my daughter's teachers said, I'm fluffy. Like she had some padding on her, and so I noticed that when we would go visit her at Christmas, every Christmas she would eat less and less, and she was just like disappearing on us, And I don't know what that is, but have you seen that in some of your older friends or loved ones?
I'm definitely seeing it with my in-laws who seem to eat less and less, who seem to avoid protein more and more. I see a bit of it with my mom, who also seems to be eating less and who's avoiding protein a lot. Like if I give her, you know, a 3.5 ounce piece of protein, for sure she's going to take some part of it off if she's here at our house and then try to give it to one of my kids. But if I offer her a sweet treat of some kind, she's going to eat that. But she doesn't seem to get the same issues with digestion when she's taking in carbs. So I'm sure that there's something there that has to do with digestive enzymes. I'd bet a lot of money there. And with my in-laws as well, I think they're really tending to go towards more with carbohydrates. And I do think it has to do with being able to digest the meat and digest the fat.
Well, just another little minor issue is I think we've had some list mates who've said that when they were high oxalate, they were getting kind of stiff. And certainly you've noticed older people as their movement gets kind of stiff sometimes. And that may be because they, you know, it's just like cholesterol building up in your blood vessels. They've had longer for it to accumulate and to cause problems. So, so there's a lot to learn by, and I want to say that the reason that we do what we do is that by comparing our stories and knowing, you know, I did this and then that happened, we can learn things that can help, you know, kind of gently nudge the research forward, because the scientists aren't going to study something that they don't know anything about, or they haven't heard a reason to study something. And so we want to be kind of in the middle there in encouraging the research to go on and to encourage its appropriate application to what goes on with you when you go to the doctor's office. So we want both to happen.
Well, and while I'm thinking of it, because we've been talking about it, we know that oxalate bioaccumulates. And I know that when I started this diet, my digestion was terrible. And I had to take handfuls of digestive enzymes to try and get my meals down. So it does make me wonder if part of what's happening is not so much that digestion gets worse as we get older, but maybe as we get older, if we've been eating oxalate and we've bioaccumulated it, maybe that's why they're not digesting as well. And I can say for sure that my mom has definitely gone off her low oxalate diet, and that we know she's got a kidney stone now, but she doesn't remember the tenets, so it's a challenge to be able to help her with her diet that way. But you know, it's just one of the things that came to mind as we were talking about people who are getting older and having difficulties with digestion. It could be related to oxalate as well, especially if you've been eating higher oxalate foods for a lifetime, and now you're in your 80s or your 90s.
Well, I also want to point out that these transporters that I was talking about that exchange sulfate for oxalate, sulfate is a huge on-off switch for hormones like gastrointestinal hormones. The gastrointestinal tract has all kinds of regulation that uses sulfate. So it might be that oxalate becomes a disruptor of some of that process. And again, you know, we certainly know the science behind what could be happening, but, you know, maybe there's not as clear science to show that this is exactly what's causing these problems, you know, and so we look on the scientists to do their job to try to clarify those issues for the rest of us.
Absolutely. Well, we've been at it for an hour, a little bit.
Yes. And it's getting late for Patricia over there in Belgium. What time is it now, Patricia?
It's 9:15.
Yeah. All right. So it's time for us to wind up.
She told us that, you know, because she's in Europe and, you know, everybody's trying to conserve fuel and all of that sort of thing. So you can see her all wrapped up there at home because they're trying to be smart, right?
Responsible. Yeah.
Responsible.
That's right. That's right.
So anyway, we wish you a warm winter.
And we wish you, the two beautiful Americans, we wish you a wonderful Thanksgiving holiday. We hope you enjoy your family and you enjoy everything that is good, that was good for this year, and our best wishes for everything that is good for the next.
Thank you.
Yes, absolutely. Us Canadians have done Thanksgiving, so we're a little out ahead of you, but all the best for Thanksgiving in the US and for anybody who's tuning us in later.
Well, look at me now, because this is me before Thanksgiving and the feast, and I'll probably be bigger like 2 or 3 days from now.
Oh, well, there is a
Enjoy
time for
yourselves,
feasting.
be grateful. I am so grateful to have you ladies in my life, and we will see you then. Well, actually, I will not be there next Monday, but my 3 co-wizards will be for another discussion of the Wizards of Oxalate.
We can hold down the fort for you, Patricia.
I am sure you can. So take care and have a lovely week, a lovely Thanksgiving, and see you next week then.